record. Is it recording? Do you get the sign?
Yep.
Yeah. Okay. Hello, everyone. Welcome to this first attempt from the Global Ranavirus Consortium to present Ranavirus. So just a few words. The GRC is basically a global community that assembles everyone interested in Ranavirus. It facilitates communication, collaboration among scientists, veterinarians, and other interested people in Ranavirus. We encourage everybody to join our GRC if you're interested. But right now, I will let Carrie to present her organization.
Thank you, Jacques. So welcome, everyone, and thank you for joining us. This is a collaboration with the partners of Amphibian and Reptile Conservation, aka PARC, our National Disease Task Team, and the Global Ranavirus Consortium. So you're attending today's webinar on Ranavirus, a threat to aquatic wildlife. So my name is Carrie Wickstead, and I'm with the Association of Fish and Wildlife Agencies, and I'm the state agencies coordinator for PARC. And I'll be behind the scenes helping to manage tech and also working to monitor the chat with Dee Olson, who is also part of our PARC Disease Task Team. We're pleased to kick off today's webinar, which is part of PARC's Disease Task Team webinar series. And our goal with the webinar series is to connect science and management and action by providing information to land and resource managers so we can help with input and feedback and spreading educational awareness. And as Jacques mentioned, today's webinar is co-hosted with the Global Ranavirus Consortium, so we're really excited to partner with them today. Before I introduce today's speakers, a little housekeeping. You'll see that we have a live transcript that has been enabled. So if you don't see that, if you scroll to the bottom of your screen, there should be a button that says CC and show captions. So you'll have that to use as live captioning for the event. And please enter all of your questions into the chat. As I mentioned, Dee Olson and I will be monitoring the chat. Between each of the 3 mini segments of the presentation, if we have enough time, we'll break for some quick questions relating to that. And if not, we'll try to save all those questions to the end. But Dee and I will be adding them to a document, so we will be getting to all of your questions, at least at the end of the presentation. So if you're not familiar with the PARC Disease Task Team, we're all about facilitating and guiding communication and collaboration on herpetofaunal diseases among our different PARC regions, federal, state agencies, and other partners. And our 2 current co-chairs are Lisa Schender with the National Park Service and Jenna Palmisano with University of Central Florida. And so we're really happy to have them. And also, if you're not familiar with our disease task team, I'll share a link in the chat. We have a variety of projects, including some former webinars that have been done by other partners. We have herp disease specific fact sheets and other resources. And very soon we will also have an updated decontamination protocol video up on our website. So as I mentioned, today's webinar is going to be in 3 parts. We're going to have a molecular and an immunological point of view of Ranavirus kicking us off with Dr. Greg Chichar. And then after that, we will have a presentation on Ranavirus ecology with Dr. Jesse Bruner. And then finally, we're going to jump into Ranaviruses in the field, detection and biosafety, presented by Dr. Joseph Milovic. So without further ado, I am going to kick it off and ask Dr. Chichar to start us off. He has a PhD in virology from Indiana University and has conducted his postdoc research associate at St. Jude's Children's Research Hospital. At the hospital, he was introduced to frog virus 3 and in 1981, and from that date until now, he's retained interest in frog virus 3 and the family to which it is a member. So we're really excited to have him here and to start our presentation. So thank you.
Thank you, Carrie. Let me, I'm not being able to page down. There it goes. Okay. So what I'm going to do this morning is basically talk about what's a ranavirus, how they replicate, how they trigger disease, and what role immunity plays. And then in the second part of this presentation, Jesse and Joe will be talking about viral ecology and viral diagnostics. So ranaviruses are large icosahedral DNA-containing viruses. They encode between 100 and 150 proteins. If you look at a model of a ranavirus particle, they're these large icosahedrons. Most of the protein is a major capsid protein, and that's this protein here in yellow. There are smaller amounts of minor capsid proteins called pentons at the vertices, and this blue is a zip protein. If you were to dissect the virus particle, you see something like this. Major capsid protein is on the outside. There's a DNA protein core on the inside that we really don't know how well it's organized. And then there's an internal viral membrane. And this internal viral membrane is rather interesting because it's found in a large number of large DNA-containing viruses, and it allows taxonomically to put them into kind of one specific phyla. If you look at a transmission electron microscope of ranavirus particles, that's shown here on the left-hand side, particles, the virus forms as two forms. One's a naked form, and the second is an envelope form. Both of these forms are infectious, and that's rather odd. Most viruses that are enveloped, if they lose the envelope, they lose infectivity, but not the ranaviruses. However, the envelope form appears to have a higher specific infectivity than the naked form. It's thought that maybe the naked form might be more stable in the environment. The envelope form might be more transmissible inside an infectious host. And this panel on the right here shows a cryo-electron micrograph that kind of gives you almost a three-dimensional view. So A shows a complete virus particle. B is up here where the capsid is partially eroded. If you look at C, the capsid is mostly gone. And what you can see is that large DNA protein core on the inside of the particle. Okay. If you look at the viral genome, the genome contains between 100 and 150 proteins. And the one thing you're struck by is the fact that the genes are very closely packed. Unlike mammals or eukaryotes, where there's a lot of introns and exons and genes become very large, the virus packs all of its information in a very small package. And the other point I want to make, which sounds a little bit kind of molecular biology-like, is that the control regions are thought to lie just upstream from a given gene. Well, let me explain why that's important. Viral genes are divided basically into different temporal classes, early and late. And the genes are not clustered by their temporal class. So what makes a gene early and late lies right upstream from it. And that means that the genes could be jumbled around in different species. And I'll show you later on how that allows us to speciate the genus Ranavirus. If you ask what's in the genome, there are two kinds of genes. There are essential genes and non-essential genes. What makes up the two categories? Well, the essential genes are pretty obvious. These include structural genes like the major capsid protein and some of the minor proteins I've already alluded to, and then a series of essential catalytic genes like the viral DNA polymerase that allows the virus to replicate its genome, and the viral RNA polymerase that allows it to produce late viral messages. The reason these are essential genes is that if you knock them down or knock them out, nothing happens. Virus replication grinds to a halt. There's a second category of genes that I've termed non-essential genes, and that's a bit of a misnomer, and I'll tell you why in a minute. So the non-essential genes fall into two categories: efficiency accessory genes and immune evasion genes. If you look at the first category, these accessory genes are genes like thymidine kinase, DUTPase, and a variety of others that are involved in nucleotide metabolism. In a normal healthy cell that's actively growing, you could knock these genes out and the virus replicates just fine. But in cells that are quiescent, they're absolutely required for infectivity. And so it turns out that although viruses lacking these genes can replicate in most tissue culture cells, if you try to infect an animal, virus replication is stopped. So they're non-essential in vitro, but they're required in vivo infection. As for the viral immune evasion genes, there are a variety of these, and I listed three of them here. VCARD, which blocks apoptosis, a beta-hydroxysteroid dehydrogenase that suppresses immunity, and then a gene called VIF2 alpha that maintains viral protein synthesis in the face of the shutoff of host protein synthesis. It turns out that, again, you could knock these genes out and the virus will replicate in most cells in culture perfectly fine. But if you knock them out and infect an animal, the replication is diminished and the virulence is reduced. And so again, they're non-essential in vitro, but they're required in vivo infections. I'll talk more about these at the end of the talk. So taxonomy of the virus Iridoviridae. The family is called Iridoviridae, and the name comes from the fact that the early viruses that were isolated were isolated in insects. And it turns out that the virus replicates to such a large extent that the insects basically change color. They become iridescent. In fact, some of you might have seen there was a YouTube thing on CNN several months ago of a guy with a bunch of blue pill bugs in his hand. And these are animals that when they became infected, the virus concentration reaches such of a level that the light refracts and it gives the insect a kind of an iridescent appearance. And if you actually purify the virus particles, the purified particles have an iridescence about them. So two families of viruses, one that affect insects that I'll say nothing more about, and a second subfamily that affects cold-blooded vertebrates. And those are the ranaviruses, the megalocytoviruses, and the lymphocysty viruses. Now, how do you distinguish one from the other? It turns out they're distinguished in a very simple way by host range, genome size, and pathology. Lymphocystis virus and megalocytoviruses viruses only infect fish, whereas ranaviruses infect not only amphibians, but also fish and reptiles. Viruses also differ by genome size and GC content. Lymphocystis viruses are much larger than the megalocytoviruses and the ranaviruses, and they have a lower content of GC residues. And lastly, they're distinguished by pathology. A fish infected with lymphocystis disease virus develops these wart-like lesions on their outer surface and probably on internal organs too. Whereas fish and reptiles and amphibians that are infected by ranavirus and megalocytoviruses developed a systemic disease that involves a lot of internal hemorrhages of internal organs, petechial hemorrhages on the skin sloughing, and a variety of other things. So there's clearly differences in host range, genome size, and pathology among these three genera. What's also turned out to be the case, and again here, the bottom line here is the top line. It turns out that analysis of gene sequences and development of phylogenetic trees has replaced physical characteristics like the ones I just mentioned when classifying viruses. And so one could do this by looking at individual proteins like the major capsid protein or the viral DNA polymerase, or a conserved set of core genes. And it turns out that among all iridoviruses, there are approximately 26 genes found in all these viruses. And you could concatenate that set, perform a multiple alignment, and then using a variety of different techniques, develop phylogenetic trees. And what do those phylogenetic trees show? That's shown here in the next slide. Phylogenetic tree using 21 of these conserved genes clearly divides the family into two subfamilies. The insect viruses, the beta Iridoviridae, are down here. The vertebrate viruses are here. And you could also see that the three genera we defined on physical features are also separated phylogenetically. These are the megalocytoviruses, these are the lymphocystis viruses, and these are the ranaviruses. Now, if you look at the ranaviruses, you can clearly see three demarcations. This is Singapore grouper iridovirus, this is largemouth bass virus and its relatives, and a variety of closely packed viruses. And so we might ask the question, within the genus Ranavirus, how many viral species are there? And you may think this might depend if you're a lumper or a splitter. And so if you're a lumper, you might say, well, you got three. You have Singapore grouper, lymphocystis, largemouth bass virus, and then a bunch of other viruses. But actually, it's more important than that. And I'll give you one example. Epizootic hematopoietic necrosis virus, which is a mouthful, is a virus found in Australia, and it affects redfin perch and rainbow trout. It's a OIE reportable disease, and so you can't ship fish from Australia unless you certify them to be EHN-free. ECV is European catfish virus. If European catfish virus is not the same as EHNV, you can't ship virus from Australia to Europe without first checking it. If ECV is the same species, but simply a different strain or isolate, you can. And so it makes a difference from commercial points of view, whether you have one species here or multiple species. And I'll give you kind of a hint of what the answer is. You basically could look at multiple species based on a variety of other criteria. And that's shown in the next couple slides. So what defines Ranavirus species? One is sequence similarity. I've clearly showed you that grouper viruses and largemouth bass virus differ markedly from the other viruses, but there's a series of viruses that show greater than 95% identity. How do you break it down after that? You could look at host specificity or preference. And so among these viruses, you could distinguish viruses that affect only fish, only anurans, or primarily caudate amphibians like salamanders. You could also look at genome size. Frogvirus 3 tends to be the smallest in the family, EHNV somewhere intermediate, Singapore grouper iridovirus somewhat larger. And lastly, you could look at, not lastly, but penultimately, you could look at gene order or arrangements. Again, ranaviruses have the same genes, but because of the fact that they could be mixed around, they are arranged differently in different species. And that's shown kind of graphically in the next slide. This is called the dot plot. And it's a little bit complicated, but it makes it very clear here. STIV is soft-shell turtle iridovirus. If you line up the sequences, the genome of STIV on a line and compare it to itself and put a dot every time you have the same nucleotide, all the nucleotides fall on a straight line. If you're comparing STIV to Frogvirus 3, you see the same thing. And what that's telling you is that soft-shell turtle virus and Frogvirus 3 are probably just isolates or strains of the same species, which happens to be Frogvirus 3. If, on the other hand, you compare soft-shell turtle virus to Amphibian tigrinum virus, and that's a virus that affects salamanders, what you see are large areas where the genes have the same order, but also where they're flipped around. And so these inversions, if you look at the gene organization in different ranaviruses, different gene organizations define different species. And the last one down here simply compares STIV to Singapore grouper iridovirus. Again, the same genes are there, but they're completely jumbled around. So gene order will also allow us to speciate members in the genus Ranavirus. The last way is what we've talked about before, and that's looking at phylogenetic trees. And this is a phylogenetic tree based on 26 conserved genes within the genus Ranavirus. And what you could clearly see again here is that by performing a phylogenetic analysis, you could define different species. Frogvirus 3, common midwife toad virus. This is European North Atlantic Ranavirus, Amphibian tigrinovirus, and EHNV. And so by a variety of criteria, we could divide the genus Ranavirus into 7 distinct species. And with the realization that each one of these species likely has a variety of isolates and strains that are found within it. Last 2 slides about taxonomy, simply I wanted to point out kind of some changes here. So if you've taken college biology or high school biology, you might have remembered the mnemonic, King Peter comes in good stock. And so the International Committee on Taxonomy of Viruses has decided that viruses should be classified the same way as plants and animals, by kingdoms, phylums, classes, orders, and families. And I simply wanted to point out that the iridoviruses, to which Frogvirus 3 belongs, are now part of a phylum which have the close to unpronounceable name Nucleocytoplasma verrucoda. And these are large DNA viruses that replicate either in or in both the nucleus or the cytoplasm. And they include things like pox viruses, African swine fever virus that affects pigs and insects, Facadna viruses that impact algae, a variety of viruses like the Mimi viruses, Pithoviruses, Marseille virus that affect amphibians, that affect amoeba, and in some cases have genomes that are close to 2,000 kilobase pairs. And finally, the iridoviruses and ascoviruses that infect insects. And the last point I want to mention in terms of taxonomy is nomenclature. Part of what the ICTV did was decide that viruses should be named by binomial nomenclature. And so what you may be seeing in the future is something like frog virus 3 won't be frog virus 3 anymore. It's now part of a species called Ranavirus Ronid1, where the first word is derived from the genus and the epithet is derived from probably the principal or the primary or the first host in which the virus was isolated. I simply want to point out that if the name Ranavirus Ronid1 sticks, it doesn't mean that you still can't refer to frog virus 3 or tiger frog virus or soft-shell turtle virus, because these are all strains or isolates of this virus species. Okay, so with that kind of done, I just wanted to go on to talk about how the virus replicates and give you a very brief description of that. Virus replication begins in the nucleus. And in the nucleus, two things happen. And I also want to point out, first of all, that both envelope viruses and, again, naked viruses are infectious, and that a wide variety of cells can be infected. So despite the fact that I've done most of my work with frog virus 3 will replicate in fish cells, amphibian cells, reptilian cells. It'll also replicate in mammalian cells if you keep the temperature below 30 degrees centigrade. So the thought is simply this. Naked viruses interact with the plasma membrane and inject their viral core into the cytoplasm, where it makes its way to the nucleus. Envelope viruses are picked up by receptor-mediated endocytosis, and either the virus particle or the core makes its way to the nucleus. In either case, viral DNA is injected into the nucleus, and two events happen. The first event is early transcription, and in early transcription, host RNA polymerase II and one or more virion-associated transcriptional transactivators collaborate to generate basically early viral proteins. And these early viral proteins code for things like the viral DNA polymerase and viral RNA polymerase. Once this viral DNA polymerase is made, it binds to the genome that's been injected, and first stage DNA synthesis takes place. And in first stage DNA synthesis, a unit-sized copy of the viral genome is replicated. So again, replication begins in the nucleus, early transcripts are made, and some of those early transcripts participate then in the first round of DNA synthesis that generates unit-sized molecules. But replication finishes in the cytoplasm. And what happens here is that the DNA that's made in the nucleus is transported to the cytoplasm. And in association with early proteins, it begins to form a viral assembly site. And the viral assembly site is an interesting structure. I'll show an EM of it in a few minutes. But the assembly site is the site of second stage DNA synthesis, in which the viral genome is formed into a large multi-branch concatemer. And it's also the site of late viral transcription, and it's also the site of capsid formation. So you've got concatemer formation taking place, you've got late transcription taking place, and you've got virus assembly taking place. And so what happens is late transcription takes place. These late proteins, these late transcripts encode for late proteins, which are primarily structural. Those proteins are concentrated inside the nucleus, and then in a process that's not very clearly understood, the viral DNA is packaged into virus particles, and you'll see inside assembly sites completed particles. You'll also see particles in things called paracrystalline arrays. And in some cases, virus particles will be budding from the plasma membrane and acquiring an envelope. And let me show you what that looks like in real life. Oops, sorry. So the panel on the left here is a transmission EM of a frog virus 3 infected cell. And you can see the virus nucleus off to one side. These darkened material represents chromatin condensation and is a stage in apoptosis. The assembly site is shown here. And what's unique about the assembly sites is they're relatively electron-lucid structures. The rest of the cytoplasm is relatively dark. The assembly site simply shows virus particles in various stages of assembly. You can also see, in many cases, mitochondria and filaments surrounding it. It's not clear whether the mitochondria are there to provide energy or simply as the assembly site expands, all these other structures are pushed off to the side. You could see these paracrystalline arrays I talked about in the cytoplasm. In fact, in some infected cells, the entire cytoplasm is filled up with these paracrystalline arrays. And then a small number of particles here are budding from the plasma membrane. In the other panels shown here, this represents the assembly process. So A1, A2, and A5 represent stages in virus assembly. If you look in the panel here on the left, and I've kind of cut it off on my thing here, the process involves membranes that are generated from the ER. They serve as a nucleation point. And what happens from these small membranes, it's thought that major capsid protein accumulates on one surface. A myristylated membrane accumulates on the inner surface. And then in time, this structure then begins to take the shape of an icosahedron. At some point, and it's not clear how, viral DNA enters the structure to form either empty particles, in the case it wouldn't be an empty particle, or a complete virus particle. Whether the capsid simply surrounds the DNA and picks up the DNA genome, or whether there's a portal in which the DNA enters is not really clear. With other large DNA viruses, both mechanisms seem to be possible. It's not clear what happens in the case of the ranaviruses. This slide just simply shows an example of the budding process. Again, many particles are present in these paracrystalline arrays, but at times particles will then bud from the surface. And it's not known, again, what drives the budding process versus staying in the infected cell, where these particles are released when the cell simply lyses. So lastly, what I kind of want to talk about is how do ranaviruses cause disease? And it's really a multi-step process. The process I outlined in the context of how the virus replicates, while the virus is doing its bit, what it's also doing is it's shutting off host cell DNA, RNA, protein synthesis. And that shut off of host macromolecular synthesis, along with basically perturbations in the membrane, leads to cell death. And that's probably necrotic cell death. So cell death can be triggered by necrosis, which is probably a consequence of the inhibition of host macromolecular synthesis. It's also triggered by apoptosis, which is a self-destruct mechanism. Again, if you think back to the old Star Trek shows, before the Klingons capture the Enterprise, they want to go through the self-destruct mechanism. And so a virus that undergoes apoptotic cell death will reduce the amount of virus production in it. And so that's a phenomenon that happens in an infected cell. What also may trigger disease in a ranavirus infection is the immune system itself. The immune system mounts a response against the infection. And like you see with many mammalian infections, this may lead to a cytokine storm or immune-mediated disease. I think many of you are aware of the fact that in the case of COVID-19, a lot of the pathology is due not so much due to virus replication per se, but to the immune response to the virus. So this combination of inhibition of cell macromolecular synthesis, the induction of necrotic and apoptotic death, the cytokine storm, all these things lead to disease. And what that results in is a variety of manifestations. So you see petechial hemorrhages on the outer surface of infected frogs and fish. You see hemorrhaging of internal organs. You see skin sloughing, and all these things lead to morbidity and, in many cases, mortality. So how does an amphibian or a fish protect itself against ranavirus disease? What a number of my colleagues at the University of Mississippi have shown is that immune responses in fish are very similar to immune responses in higher organisms. And that's true both in fish and, as Jacques Rivera showed, in amphibians. So we have antimicrobial peptides that either block virus, either inactivate virus directly or potentiate an immune response. We have the production of a variety of interferon types, type 1 and type 3. Type 1 seems to protect adults. Type 3 is produced by larvae, but is not as protective. Infected animals produce antiviral antibodies that neutralize infectivity. And there are also a variety of cytotoxic cells that will kill virus-infected cells and limit virus replication. And these cytotoxic cells can be both innate cells like natural killer cells or they could be result from acquired immunity, such as CD8 positive cytotoxic T cells. So this combination of antimicrobial peptides, a variety of interferons, antiviral antibodies, and cytotoxic cells basically allows animals to survive infection. And the surprising thing is that adults are relatively resistant to MP3, whereas tadpoles, metamorphs, and immune-suppressed adult animals are not. And so one sees infection, but not a lot of mortality in adults. One sees morbidity and mortality in these other classes of amphibians. And the last thing I want to mention is viral vaccines. So, one thing you might be concerned about is how can you protect these animals before they become infected? And it's been shown that a variety of inactivated and recombinant protein viral vaccines provide protection against megalocytoviruses and ranaviruses. And this has been shown in the case of red seabream iridovirus, which is a megalocytovirus, a virus that infects the giant Chinese salamander called ADRV, and protection of grouper by Singapore grouper iridoviruses. These are all, if you have high value animals that are large size, you could actually, it's cost effective to inject them with these vaccines and protect them from infection. Another way to do it might be by immersion. And what Jacques Robert and his colleagues have shown is that if you knock out key genes in frog virus 3, the virus will still replicate in vitro. And that's where I was talking about essential versus non-essential genes. But these knockouts show reduced replication and virulence in vivo. And let me just give you one example here. If you take tadpoles and you infect them with wild-type virus, that's these open circles, or with 2 of these knockouts are in the VCAR gene and in the beta-hydroxysteroid dehydrogenase gene, what happens? By 30 days post-infection, about 80% of the animals exposed to wild-type virus have died, but only about 20% that have been exposed to either the VCAR knockout or the beta-hydroxy knockout have died. And so reducing or removing these genes by knockout allows you to generate viruses that show markedly less morbidity and mortality in vivo. And it's hoped that then these knockout viruses might be used as attenuated viruses to protect these animals. This has not been done for the present time, but it's something that clearly could be possible. If you could mass expose a variety of young animals to these viruses by immersion and hopefully protect them from a disease later on. And with that, I'm basically finished. I'd like to acknowledge my collaborators. And if there's any time, open the talk up for questions. Carrie, I think it goes back to you now.
Yes, thank you, Greg. So we do have at least one question in the chat. The first question is from Dee Dee Olson, and she wants to know, are there microbial properties against RV from skin microbiota as well as hosts?
Yeah, actually, we'd shown a while ago, collaborating with Louise Rollins-Smith, that a variety of amphibian-derived antimicrobial peptides will inactivate frog virus 3. Whether or not they also potentiate the immune system, I'm not so sure whether that happens. We did not study that.
Thank you. And Jonathan Colby also asked, could ranaviral vaccine bats, bats in quotes, possibly help reduce RV prevalence in industrial scale bullfrog farms?
Yeah, that's a good question. And that's actually one areas where they could actually really make a difference. I mean, I know that in the large scale bullfrog problems, they've had problems with virus outbreaks. And so one could imagine that infecting tadpoles with one of these knockout viruses might protect the tadpoles as they get larger. Actually, Jacques maybe better answer that question because he was the one who did the challenge assays. But that would be a real benefit in animals like that.
Jacques, do you have any follow-up there?
Not a lot. I mean, again, yeah, it's possible that immunizing tadpole may have memory in adults, but we have not shown that yet.
Thank you. And then we have another question: how many copies of DNA in general packaged on a particle?
We think it's just one. And I say I think because we've not really done the studies. Trying to resolve how you pull off just one virus particle from that large concatenary is not really clear. The model is based on what happens in some of the bacteriophage that replicate by the same mechanism, but it's thought that there's just one copy of viral DNA in each virus particle.
Thank you. And we've got a lot of questions rolling in. So the next question is, are any potential explanations why larvae positive metamorphs are more impacted by ranavirus than adults?
Jacques, could I pass that question to you, since you're kind of more the amphibian guy than I am? I don't know if you could hear that or not.
So yeah, again, we think tadpoles have an immune system a bit less. It's still under development, so it's a bit more immature. And in doing metamorphosis, is even more weaker immune system because everything is remodeled during this time. So there's a lot of like T cell that dies for a new differentiation of T cells. So it's a bit a moment that is more weaker in terms of infection.
And Eric asks, do we know what specifically aids in the intraclass jump in frog virus 3? I've read that US22 genes are speculated to aid in that.
Actually, the answer is no. I mean, what's interesting is that you find a given viral species preferentially will infect one host, but will infect other hosts with lower amounts, with lower efficiencies. But then you also could find that they jump they could also jump class, and we don't really know what determines that. We don't know whether it's changes in the capsid protein that give it a bigger host range, or whether the proteins that interact with cellular proteins that play a part in replication. That's just kind of an unknown study right now. If I could kind of make a plea, I mean, the problem is hard. It's been hard finding funding in the U.S. to kind of do these molecular studies, and those are the kind of studies that really need to be done. Most of the current research on the function of ranavirus genes and megalocytovirus genes is being done in Asia, and it probably reflects the fact that they have a large mariculture industry there, and ranaviruses cause significant problems in those regions.
Right. Thanks. And last but not least, before we hand it over to Jesse, so are protections only for frog virus 3 or other RV strains?
I don't know. I don't think we've never tested.
Yeah.
There is a fair amount of sequence, like among the major capsid protein, there's a fair amount of sequence conservation. And it would be interesting basically to immunize with one ranavirus and see how much it carries over. If protection is basically mediated by a response against a major capsid protein, you may get a lot of cross protection. If it's another protein that plays a role, it may be lower. But that's a really good point.
All right. And Eric just wanted to follow up that they're trying on their end with looking at the intraclass jumps. So
yes.
All right. Thank you so much, Craig. Really appreciate it. And if you could stop sharing, we will let Jesse share his screen.
Right now. Okay.
All right. And while Jesse's getting set up, we're going to have Dr. Jesse Bruner, who has a PhD in biology from Arizona State University, speak about ranavirus ecology. And he has worked on the ecology of ATV and tiger salamanders. And from there, he spent his postdoc at the Carey Institute of Ecosystem Studies, where he worked on tick-borne disease ecology. So currently, Jesse is a professor at Washington State University, where his lab group works on all aspects of the evolutionary ecology of pathogens, especially ranaviruses. Thank you, Jesse.
Hi, thanks. I hope everyone can hear me. So I'm going to try to hit on a number of topics fairly quickly and shallowly. So I'm going to give you like really broad brush kinds of perspectives. So I wanted to start with just the host range of the different viruses. So this is the tree that Greg just showed. And And I wanted to show you that, broadly speaking, frog virus 3 affects bony fish, amphibians, and reptiles. CMTV has likewise been found in each of these, although much more commonly in amphibians. The ENAR and EHNV have just been found in fish. And ATV so far is a bit unique in that it seems to infect primarily chaudates, primarily salamanders, although there's a little bit of evidence that it does infect frogs as well. But importantly, I just want to point out that host range has not been well established. This largely reflects the species that these viruses have been pulled out of or isolated from, and not necessarily experimental evidence about the potential host range, like who could be infected. And then where do we find these different viruses? The important point here is that basically all of these viruses are found all around the world. And so you can see FE3 in fish has been found North America and Europe and Australia. I haven't seen this in Southeast Asia, but that is probably true too. And the Santee Cooper Ranavirus, or the largemouth bass virus that Greg was talking about, has also been found all over the place. And a lot of this really depends on how you define things. So that is fish, and you'll notice it largely reflects where are we looking at fish for viruses. For amphibians, I think in large part because people have been looking at amphibians and amphibian disease around the world, the distribution is a little bit broader. FV3, the type virus, has been found essentially everywhere. CMTV has been found more recently, and I think we're finding it more and more often. And again, ATV is a bit unique in that it's only found in Western North America. But importantly, there are a lot more ranaviruses that have been found that haven't been differentiated or determined down to species. So if you look at a map of actual isolates of ranaviruses that have been found, it's much more filled in than this. And then lastly, reptiles, again, we've got FE3 and CMTV that have been found. And really what we're seeing here is just where we've been looking for pathogens, especially viral pathogens in reptiles. We really just don't know that much. So this map doesn't necessarily represent a restricted distribution, but just restricted knowledge about where those viruses are found. Okay, so moving on, we're going to talk a lot about the ecology, especially things like transmission. And one thing that we need to start with is just recognizing that the dose, the amount of virus to which an animal is exposed, is really critical for almost everything that happens. So dose affects very strongly the probability of infection. So this is like one of my own dose response studies from graduate school where I was exposing salamanders to increasing doses of ATV, and you'll see that the probability or the proportion infected increases as a function of dose, which is really, really common. So dose affects probability of infection, the probability of mortality, how quickly those animals die, and probably how capable those infected animals are of passing on the infection. Presumably, if they have more intense infections, so higher viral titers inside of them, they're more able to pass on that infection. Okay, so how does the virus actually get between hosts? Well, really, there's actually a wide range of routes of transmission. So it can be passed through the water. It can be passed between animals by direct contact, including this little 1 second belly-to-belly contact. Certainly by consumption, and this ends up being important in a lot of animals. Even, presumably vegetarian tadpoles often will prey on slower moving or sicker tadpoles. So it can be predation and cannibalism, but also necrophagy. I want to point out that there has been some sort of circumstantial evidence that perhaps mosquitoes might be moving ranavirus, but this has never really been demonstrated. And I think we could probably cross this one off for right now. Anyway, there's, at least in the lab, there's a lot of ways to move virus from one host to another. How does this work in nature? Well, I think the question really boils down to which of these routes or which paths transfer enough virus to a naive host to cause an infection. And so I think in aquatic species or in aquatic stages, this is primarily through direct contact Definitely through the water, but if we're willing to assume that these animals live in large bodies of water and that the water is relatively well mixed, we start to run into problems with dilution. So when you move further and further from an infected animal, there's less and less virus in the water, meaning less and less chance of infection. So I kind of think about these as more or less the same thing. When animals are in close contact, whether they're directly touching or just near each other, there's the potential for transmission. Consumption can be really important, and especially some theoretical models suggest that this is especially important during a die-off, but maybe not early on. I should point out, though, that consumption is probably a really good way to get infection from one type of one host species to another host species where they don't interact very often. In terrestrial stages and species, we honestly really don't know because they're really hard to watch, but presumably direct contact and consumption are going to be important. So transmission between taxa, I should point out in laboratory experiments, waterborne transmission has been demonstrated. You can transmit infections from a turtle to a frog, or a frog to a turtle, or a fish to turtles, etc. But again, I just want to point out that we sort of have the same problem with dilution. So transmission through the water to some non-target species is probably much more likely during a big epidemic. My suspicion is that trophic transmission, so predation and necrophagy, is more important than transmission through the water. And this is largely because it has been observed. We've seen, for instance, a snake, a natrix, that was eating infected newts, I think it was, and it was infected with the virus from that newt. And often what we see is in nature, we don't exactly know who got who infected, but we'll see an epidemic in amphibian, especially amphibian larvae, followed by mortality in reptiles. So this, again, is not well known, but this seems to be a broad pattern. So ranaviruses are broadly thought to be fairly robust in the environment or to environmental perturbations. They can certainly persist in the water. They can persist in soil or pond substrates. They can persist in infected carcasses as well. This is important. A carcass itself can be infectious to others. But those viruses do degrade, and they degrade faster at higher temperatures, as you might expect. If they're dried out, at least in pond substrates, they seem to be inactivated. And microbes and zooplankton can also degrade the virus. So there's some evidence that in the summers, when temperatures are hot and there's lots of microbes and zooplankton, ranavirus may not persist terribly long, at least in an active form. Ranavirus does certainly persist in hosts between epidemics. So there's evidence of persistent sublethal infections. Some of this comes from Jacques' lab, where they've demonstrated quiescent infections in macrophages for months in xenopiths. And then some of my own work this provides more circumstantial evidence, but we found both tiger salamanders and wood frogs returning to ponds to breed in the spring, and they're carrying low-level ranavirus infections, and presumably they're able to restart infections or restart epidemics that way. So persistence between epidemics is a really different question. Ranavirus could certainly persist in carcasses. It could persist in frozen waters or frozen soil. It could persist in hosts between epidemics, especially in the winter. So I just want to point out that there's a real distinction between persistence within an epidemic and how much transmission there is from the environment versus transmission between epidemics. Okay, let's talk about the outcomes of infection. And what I'm kind of trying to lay out here graphically is that we can think about individual level outcomes and also population level outcomes. And really, there's a wide range of outcomes. So sometimes individuals are essentially refractory. They are uninfectable, or they very quickly clear infections. They can also develop sublethal or persistent infections. Most often we see moderate disease or mortality. And then they can be very, very lethal, maybe 100% lethal within days to weeks. And below this line, we can kind of see the population level consequence. This can range from essentially ranavirus disappears to we can see ranavirus sticking around, but without any mortality. We can see moderate mortality and loss of some fraction of the population. To at the extreme, we can see there have been cases where it seems like the entire larval population disappears within a day or is dead within a day. This is sort of my gut feeling about how common, these different types of outcomes are. Now, why are there such different outcomes? There's just a really wide range of possible explanations. The first thing I want to point out is that detecting mortality is difficult. So we're probably missing a lot of mortality events or low-level mortality. Carcasses disappear surprisingly rapidly. But past that, setting that aside, we do know that there are differences, or there are often differences in the virulence or the negative outcomes for populations between different viral species and strains. So for instance, there's some evidence, but it's inconsistent, that the CMTV species is more deadly than FV3. There are some recombinants that mix both of those that seem to be especially virulent, but we don't know if that's a consistent pattern. There are also certain host species that are more susceptible. Wood frogs in North America tend to be highly susceptible. Their congeners, bullfrogs, tend to be very resistant. And then there's different stages, like Greg was saying. Larval amphibians tend to be more susceptible than adults. In the fish viruses, it tends to be the juvenile fish that are much more likely to suffer mortality from ranavirus than the adults. And then lastly, environmental conditions are certainly playing a role in this. So again, back to the fish, a lot of mortality events in fish are associated with stressful events like temperature spikes or oxygen deprivation. And then there's a wide variety of possible environmental conditions associated with ranavirus-associated mortality in amphibians, but one example is that wood frogs raised in salty conditions seem to be more likely to experience die-offs. So there are few clear answers about why we get such different outcomes, but no shortage of possible explanations. I wanted to just hit on the potential for seasonality in ranavirus outbreaks. So this is I'll admit, a very complicated figure from a chapter in the Ranavirus book from 2015, which, by the way, is available free online, and we are revising that at present, so there will be a new version of this soon. Anyway, what I'm showing you here is the day of the year, and these horizontal lines represent the spring and autumn equinoxes, and this is latitude. And these blocks here basically discuss how there are different records of die-off events. And so you can see, for instance, in North America, in the wild, that's the blue here, most of the die-off events were noted in the spring and summer, but then there are exceptions. And so, again, there are lots of possible explanations. Some of this die-offs tend to be associated with temperature, elevated temperature, but also they seem to be associated with particular developmental stages right before metamorphosis. And we can't really discount that maybe mortality events happen at some time after the virus is introduced. So we would just have to wait until an epidemic hits enough of the population that then you see mortality events. Again, in fish, the die-off events are often associated with poor environmental conditions like temperature spikes in the summer. So there is clearly seasonality, but that seasonality is probably very system specific. Oh man, I blazed through this. So let me end and see if there's any questions. Oh, I thought I blazed through it. Maybe I took forever.
No, you did go through very fast, but it was very informative. So thank you, Jesse. So we have a couple questions in the chat, and I'm sure a few more are going to be filtering in as well. But the first question is, how does environmental temperature or co-infection with other pathogens affect RV infection and host response?
So temperature is undoubtedly very important for ranavirus the host pathogen interaction. The viral growth, viral replication is definitely temperature dependent in culture. Host responses seem to be temperature dependent. And in a wide variety of experiments, the outcome, the mortality tends to vary as a function of temperature. The problem is that there isn't any single consistent pattern. Sometimes there's more mortality at higher temperatures, some at lower temperatures. It probably depends on both the host and the virus and the particular temperatures. So the temperature is clearly important, but I can't give a simple rule. Co-infection, likewise, is probably very important. There has been recently increasing work showing that co-infections do occur and can, depending on the study, can seem to make infections with ranavirus more or less productive and lethal. So again, it's a bit of a mixed bag, and I think we're too early to say how co-infection changes the outcome.
All right, thank you. And then our next question is there any evidence of vertical transmission through the eggs?
So that's a great question. That's something I didn't really hit on. So there was some debate about the importance of vertical transmission. And we need to make a slight distinction. So there is true vertical transmission where the virus ends up getting inside of the egg, is like transmitted from mother to the eggs, and then those eggs have the virus inside of them. And there doesn't seem to be much evidence that that actually occurs. However, there can be what we would call pseudo-vertical transmission, which is like an egg mass can be contaminated with virus. And when say the larvae hatch out, they are then infected at that point. And there is quite a bit of evidence that actually does occur. We've I think a number of us have had problems where we brought in egg masses, like amphibian egg masses, and then had them become infected, presumably with contamination. So that does occur. How important that is in the whole epidemiology, I'm not 100% certain, but my guess would be that's a great way for infected parents to transmit the infection to the next generation.
Thank you. And Evelyn just noted that they'll be presenting later on some experimental work addressing co-infection with BD. So good plug to stay on and listen to the following presentations. So our next question is from Laura. Has anyone looked at vitamin deficiencies as weakening immune systems to make animals more susceptible at different life stages? Currently in fish, we're seeing correlations with thiamine deficiencies.
I don't know of anyone that's been looking at vitamins per se. I do know that people have looked at nutrition, and nutrition can be really important in ranavirus outcomes. Erica Crespi's lab, for instance, has been looking at algae supplementation and food quality, and it does seem to be really important. But interestingly, it can make infections more intense, but might make the animals more tolerant. But exactly how that works and how vitamins play a role, I don't know.
Interesting. Yeah, give them all some vitamin C, right?
Why not?
And there's another question on the vertical transmission. Would what you described make you think it is unlikely in reptiles?
That vertical transmission is unlikely in reptiles.
Mm-hmm.
I think my guess would be that it's less likely, but I wouldn't rule it out entirely.
Okay.
And I'm just guessing.
Sounds good. Well, thank you. I don't see, oh, there's one more from Michelle. So what about vertical transmission in live-bearing reptiles?
Does anyone in the chat know anything about vertical transmission in live-bearing reptiles? I mean, we know so little about ranavirus, especially ranavirus transmission in reptiles. So I really don't know. It's a good question.
Yeah, seems like a good research subject to look at.
There we go. If someone's looking for a dissertation, there you go. Okay, thank you.
All right, thank you. Really appreciate it. All right, last but not least, we are going to jump into our final segment of this introductory webinar. And we have Dr. Joseph Mielovic And he is an assistant professor at Northern Arizona University's School of Informatics, Computing, and Cyber Systems, or SICCS, and he's an affiliate researcher at NAU's Pathogen and Microbiome Institute. And he was having some issues with his Wi-Fi. Is he on still?
I don't see him.
He said he put the video, he pre-recorded it, so it must be in that folder. So maybe we'll just play that. If you guys could play it.
Yeah, let me pull that up.
Oh, he's here. Joe,
Yeah, sorry.
can you hear?
I went to share my screen and then it said I needed an update. So let me just do that now. Okay. Are you able to see my slides?
Yes.
Yeah.
Okay, thank you. Right. Hi, everyone. I'm Joe Mihaljevic. I'm an assistant professor at Northern Arizona University, and my research has mostly been in larval tiger salamanders out in Arizona, and we're looking at ATV and climate interactions. I'm not going to talk much about that today, but I am going to talk more about how do you go out into the field and what are the different types of methods to detect ranaviruses in the field, different samples you can take, different mechanisms to look for the virus in the lab, and then some notes on biosafety as well. So I would say that the most common workflow is what you see here, is you can go out into the field and you can collect various types of samples, and I'm going to go through what types of samples and the pros and cons. And then you typically either send those samples to a lab, or if you have your own lab, you'll bring the samples to the lab. You'll do tests to extract viral DNA, and you'll do methods to try to detect that viral DNA in the sample. Now, this isn't the only way to detect ranaviruses, so I want to go over at least some of the others and why you might do them. So one of the methods to detect ranaviruses is through what we call histology and immunohistochemistry, where you can stain tissues and try to detect the virus inside the tissues. And this is a way to confirm that a ranavirus is actually infecting the tissues in the host of interest. And this histology can also be used to determine clinical pathology, or to try to infer whether the virus has actually killed an organism, for instance. So this is used often for if you're trying to figure out why an individual has died and you're not sure it's the virus. This is one way to help you get at that question. Another detection method is through viral isolation on cell culture, which Greg talked a little bit about. But this helps, again, confirm that a ranavirus is present in a sample. So if you have a sample like a tissue or other type of sample, you can try to isolate the virus and grow it on cells in the lab. This allows you to grow the virus for downstream applications such as genomics, which I'll talk about just in a second. Or it can help you grow the virus in the lab if you're doing experiments, for instance, and you need a larger quantity of the virus. But this, again, requires live virus in order to grow the virus on cell culture. It has to replicate in those cells. Now, the most common method, if you're just trying to detect the presence or absence of the virus in samples, is through DNA extraction and detection of viral DNA. And so this is used to confirm that a ranavirus is present in a sample, and it can be used for downstream applications like genomics. So for this most common method, where you're trying to identify if the virus is in a sample by looking for pieces of its DNA, there are different methods for this. And these aren't the only methods, but these are probably the most common ones. So first you have conventional PCR, polymerase chain reaction. So this is looking for maybe like a 400 to 500 base pair piece of the genome of the virus. And this is used to say the presence or absence of the virus in a sample. I would say it's more quote unquote old school, but we still use it in our lab because it can be useful for downstream DNA sequencing, where you're actually sequencing the DNA, and this can help you even from like a 500 base pair part of the genome, say if that virus is more like FV3, is it more like ATV? So for species level distinction of the virus. Now, conventional PCR, however, isn't as sensitive of a test, so you can't detect very low quantities of the virus, so you might get false negatives. Now, qPCR or quantitative PCR also detects the presence or absence of the virus, but with much more sensitivity, so you're able to detect smaller amounts of the virus in the sample. And if you do this in certain ways with the standard, you're able to quantify the viral DNA copies in the sample. So this gives you a measure of titer or load. So how much virus is that individual experiencing in the sample? And I would say this is a more contemporary method. It's usually faster to do in the lab, and there are methods to make it cheaper. The other part of this, which actually combines some forms of PCR, but not always, is genomics, where you're sequencing the DNA, and this is used to determine the species or strain of ranavirus that's present in a sample. You could have various reasons for doing this. If you're doing surveillance and you find virus in a place or a species you don't expect it, you might want to know what virus species is infecting that organism or is at that location. And so you need to do some type of genomics, whether it's sequencing a small amount of the viral genome or the entire amount of the viral genome. This usually requires viral isolation prior to sequencing because you need to grow enough of the virus to have enough information to look at the whole genome. And this means that you need a live virus to grow and sequence. So we've actually sequenced directly from infected tissues, but because usually there's not a lot of virus there, it can be pretty costly to do that. So it's better to grow the virus ahead of time before doing this genomics. So you need live virus, which I'll talk about why that's important and what types of samples and storage mechanisms you need in order to make that work. Now, generally, genomics is more costly than PCR and qPCR because it gives you more information and it's more labor intensive. So again, qPCR is the typical standard for diagnostic testing. So if you're going out into the field, how do you collect samples? If you're capturing live specimen, we usually use a seine or a dip net or even hand capture depending on the sensitivity of the species. So seines are good for larval capturing, but there are some species that are really sensitive to being captured by the seine. So definitely talk to your local experts on how they would prefer you to capture organisms for testing. So what are the different types of samples that you could detect in the field or collect for detection? So you could collect tissues. This could be through destructive or lethal sampling to collect liver and kidney tissues. You could do non-destructive, non-lethal methods of detection, which involve tail clips, toe clips, swabbing, and swabbing can be have different categories like buccal swabs of post-metamorphic individuals. So you're swabbing inside the mouth, or you could swab the body or cloaca. You could also not have to collect any individuals at all and do eDNA or environmental DNA sampling through water filtration. And I'll talk a bit about the pros and cons of all of these. So which sample of all of these do you want to collect? Well, conventionally, liver, kidney, and spleen have been the gold standard. but this requires destructive sampling. So on one hand, you may have a moribund or a deceased individual that you want to see if they were infected with the ranavirus. And so in that case, euthanasia, or they're already deceased, so it's not that hard to collect these tissue samples. But if you're doing surveillance, you might not want to do destructive sampling for obvious conservation reasons. So the good news is that we're often able to detect ranavirus with non-lethal methods. And so I have some data here from Jesse's group. And on the x-axis, we have basically the number of copies of the virus in the liver and kidney, which again is kind of like the gold standard. And here we have the probability of detection. And we have toe or tail clips, swabs, or eDNA filters. And what you see here is that if you have high levels of titer inside the individuals in their liver and kidney, so you know they're infected and they're highly infected, you're most likely going to be able to detect that through any of these methods. Although you can see the filters, there were some that were negative here. Although you can get some false negatives if you're using these other non-lethal methods. It's just kind of like, what are you willing to tolerate in order to use non-lethal methods? Now, there was a recent study that came out in Viruses in 2022 that actually showed or provided evidence that suggests that buccal and body swabs may be just as accurate, if not more so, than liver, kidney, and spleen samples. Now, this figure is a little hard to see. I couldn't find a better quality, but so this is in green. These bars are the proportion of true positive that were detected. Here you have liver, here you have kidney. They're a little less than 60%, which seems a bit low to me, but this is what they found in this study. Then this is comparing swabs, buccal, body swabs, and these are actually at or above 60%, which is better than the liver and kidney. So this at least provides some evidence that in these species of frogs and in post-metamorphic individuals with FB3 and CMTV, that swabs might be just as good. In the field, we've also seen, and again, this is from Emily Hall and Jesse Brunner's group, in the field, titers on water filters or eDNA are strongly correlated with the titers in host tissues. So if you have hosts at a site that are heavily infected, you're often able to detect through eDNA heavy infections at the site or high quantities of viral DNA at the site. I'm going to come back to that a little bit later as well. So if you've decided to collect tissues or swabs, how many should you collect? And it really depends on your goal. So if your goal is that you have a moribund individual or a deceased individual, and you're trying to figure out if ranavirus is present in that specific individual, then you only need to sample that individual, and you should probably collect tissues if it's already deceased. Now, if you're doing surveillance and you want to ask some of these other questions, like, is ranavirus present at a site at all? You don't know if it's there or not, and you want to collect enough samples to try to figure out if it's there. Well, maybe it's obvious, but a bigger sample size will make it more likely to detect the virus, especially if it's rare. And I have an example here from the Ranavirus book, and Jesse is working on updating that section as well. But let's say you only have 5% of individuals at a site that are positive for ranavirus, and you want to know how many samples do I have to collect at least one that's positive, and that'll tell me that it's present at the site. Well, you're looking at, if you have a large population, collecting around 60 individuals, and that's still not a guarantee that you'll find it. So you can see that that's a pretty high number of individuals and effort if ranavirus is rare at a site and only in 5%. If it's less rare, then you don't have to collect as many individuals. Now, if you're trying to quantify prevalence at the site, which is the fraction of individuals that are infected at a given time, then again, a bigger sample size will make your estimate more precise. and potentially more accurate. I wanted to make a note on filters from my own personal experience at sites in Arizona. We sample tiger salamanders in cattle stock tanks, and a lot of these sites are highly turbid. And it can actually, it seems like maybe it's not a lot of effort to collect the eDNA samples, but in our case, it can be a lot of effort to try to get this water to pass through the filters, and the filters can get clogged really quickly. And it's unclear of the impact on detection. But we often at sites that are highly turbid will rely on swabbing rather than eDNA, just in case we're unable to detect it in these clogged filters. So it's important to consult the standard methods for eDNA from the papers that I've cited earlier, and make sure you're following those protocols really well in order to maximize your chance of detecting it in the filters if present. So now you've collected samples. How do you store those samples in the field? There are two conventional methods, although I'll say there are emerging methods, there are other methods, and I'll get to that. But typically, the two methods are you could put it immediately on ice or freeze it immediately if you can, or you could put the samples in ethanol, ranging from 70% to 95% ethanol. For freezing, the pros are that this could keep the virus alive in the sample, meaning that it allows you to isolate the virus on cell line later. So if you need to do genomics, if you need to grow the virus in the lab for whatever reason, keeping it alive is important, and so freezing it is the best method here, and it preserves the DNA well. But the cons, of course, are that it may be difficult in the backcountry to do this, and freeze thaw can degrade samples. With ethanol, it's easy to prepare the ethanol and store it. It's very stable and preserves the DNA instantly. It's not really as temperature sensitive. The cons are that if you have a lot of it, you have to deal with biosafety of flammability. And if you use ethanol, it preserves the DNA of the virus, but it kills the virus. So you can't isolate the virus on cell line later. Some other small notes at the bottom here that for swabs, you can also buy a stabilization media that allows you to store the swabs in this media in the field at room-ish temperature. If you're really in a hot environment, you might want to, think about still putting it on ice. If you're doing histology, you need special protocols that involve the fixation of tissues with formalin. So definitely consult an expert in the field before you go that route, especially for biosafety. Some unsolicited advice is in working with collaborators, if genomics is necessary and we need to isolate the virus on cell line, I often suggest, if possible, collecting duplicate samples in the field. So let's say you're doing a tail clip. You might split that tail clip in half and freeze one and put one in ethanol, where I could test the one that was stored in ethanol to see if the virus is there at all. And if it is there, then I can go back to the frozen sample that's paired with that ethanol-based sample and try to isolate the virus and grow it for genomics. For filters, we typically just use ethanol or a stabilization media because you're not likely to isolate the virus from filters anyway, and you're just looking for presence or absence there. Now, I don't want to say that's a one-size-fits-all thing, but that's just some advice. And one last slide on biosafety. Of course, use gloves, just as if you were working with the chytrid fungus in the field. And more so if you're doing surveillance and you're handling multiple individuals with the idea that the virus could be at the site, you likely want to change your gloves or disinfect your gloves in between handling individuals so you're not spreading the pathogen around, and definitely disposing of gloves between sites. In terms of other equipment, including boots, you want to clean and disinfect equipment in between sites, and Molly Blutz has a really nice protocol that's been updated in 2022 but I'd also say consult your local jurisdictions to see if they have preferred or required protocols if you're working on their lands. But the key elements of any cleaning and disinfection protocol are that first, you should remove the grime from boots and equipment. As Jesse said, this pathogen can be transmitted in wet soil, in just water. And so you want to make sure that you get off all of the wet, grimy stuff. So with a brush or a power washer is nice and easy to do. And then you want to disinfect surfaces with bleach or Virkon at those recommended concentrations for at least 5-minute contact period. And so with that, I think that's all I've got, and I'll take any questions. Thank you.
Thank you so much, Joe. So we do have a few questions already in the chat. First one is from Didi, and it is, with qPCR, what factors increase uncertainty of diagnostic results, false negatives and false positives?
That's a great question. It really depends on what the sample is. So you can do qPCR, let's say on environmental DNA through filtration, those samples. And so there's a lot of environmental inhibitors that can mess with the qPCR reaction. And so there are methods to try to deal with that and get rid of the inhibitors, but they're always going to be an issue. And there are methods to try to detect if inhibition is happening, but there's not a ton of great methods to get rid of all of the inhibition in the qPCR reaction. And if you're dealing with tissues samples, it kind of depends on like how old or degraded the tissues were. Like if you had a carcass that you're trying to test from, the DNA quality could be low, and so you might not get great DNA extraction volumes. You could also put too much tissue in the reaction, and then you kind of overwhelm the reaction with host DNA. But there are ways, a good lab will try to mitigate these things. I hope that answered your question.
And the next question is relating to genomics. Is there a marker gene for viruses like there is for bacteria and fungi, so that you don't have to sequence the whole genome?
So I think you're referring to like the bacterial 16S, and like, could you detect a bunch of different viruses, like a bunch of different ranaviruses at the same time? It's kind of like yes or no. Like there are known markers that distinguish between the ranavirus types, but I wouldn't say there's like a good assay that is developed that quickly does that for you. Usually you're dealing with sequencing. Now, I would say in terms of what gene could you look at, like the major capsid protein is typically what is sequenced to tell type, but usually we refer it to like FV3-like. And the reason is that there are recombinant types out there that combine DNA from, say, FV3 and CMTV. So if you only have a small chunk of the DNA, you can say what it's most like, but not necessarily the full lineage of the sample, I'll say.
Yeah, if you can add with that, I guess the MCP, even across more distant ranavirus, the identity is very high. So it will just tell you this is a ranavirus, but you cannot use that really to tell what type of ranavirus it is. So you will have at least to do all the genes. We have discussed a bit that with some biologists trying to see if we can come with a multiple, at least, set of genes to help for distinction, but there's no consensus so far.
Okay. And the next question is, when looking to collect samples by minimally invasive methods from live specimens, have researchers used bodily fluids instead of body swabs, or bodily fluids collected alongside of body swabs? If they're two different sample types, has the accuracy of analyzing bodily fluids versus body swabs been analyzed?
It's a great question. I don't know specifically if there's been a side-by-side comparison of bodily fluids, including blood. You can detect ranavirus in blood. I don't know about other bodily fluids, but like for instance, in reptiles, blood is sometimes used to detect it. I'd say it's less common in amphibians, because let's say you're trying to detect from a larval amphibian. I know through colleagues that say that it's quite difficult to extract blood without really injuring, if not killing, the larval individual. So I don't have a great answer to that. I know you can detect it through bodily fluids, but I don't know the comparison with swabs.
Thank you. And there's some information in the chat from Jesse about additional information about disinfection guidelines, and then Dee Dee just referenced that there is a new biosecurity protocol that just came out in 2023. It's the Bletz et al. And there's also a Northeast PARC fact sheet on biosecurity that kind of echoes some of those protocols, and we've added that to the Wakelet. So
thanks for updating that and catching that. Appreciate it.
Yeah, I think the Bletz et al. Just, the recent paper just came out too, even though.
Yeah, I think I put the wrong year on that.
That's all right. And Dee Dee has a question. During surveillance, the genomics information is not always pursued. Should we be pushing for that level of analysis?
It's a great question, Dee Dee. I mean, I certainly would like to know more about, and Jesse pointed out that it's just not often done, and it's a matter of cost. And like, what does that information provide you? And sometimes it can provide you a lot of information, like maybe you didn't expect to find that type of virus in that location. It of course would give the scientific community a better understanding of the ecology of the virus and its hosts. And like Jesse said, we don't have perfect information about host range of the pathogen. And so knowing more about which types of viruses are present in which hosts would be great. And then also for co-infection, we know that there are recombinant strains out there, like FV3 mixed with CMTV in the same virus genome. But we don't know how common that is across the landscape. And we don't know if like ATV and FV3 are mixing in the same populations very much. And so those are interesting questions. Whether or not they're actionable information is not always clear, but it would certainly be actionable if you didn't expect a virus type in a certain location and it constitutes like a new emergence of the pathogen or a new translocation. That would be really important. So I would say if you're finding specimen that you don't normally see dying of virus or dying of a cause you don't know, then the genomics might be worth it.
Good to know. The next question is, what management responses are on the table for ranavirus detections or die-offs in the field? So
I know that Molly Bletz's group is working on this. They're writing a paper on that right now. I would say I'm not an expert on management, and it's tough. I don't know if there's a lot of great management in the field. I think you want to think about non-native species, like bullfrogs could, for instance, in their non-native range, could move the pathogen around others, but native species can do that too. So yeah, I don't know. Does someone else on the call have a better answer? Sounds like no.
Nope. It's difficult.
The best that we get are like reporting systems, like especially if it's not someone doing any molecular work, they might just be doing some on-foot survey and then bringing them to, we'll hear at like UF, like the vet school or to some other folks. But there's, as far as like for conservation implications, it's like you just sort of report something and not much seems to come out of that.
Yeah, I think it's hard because, you know, a die-off can happen so quickly, so you can't really manage it in the event itself. And then there's also questions on, like, if a die-off does occur, if it's not occurring every year, is it having large population effects? It's unclear. So those types of studies take a lot of time to figure out.
And Dee Dee mentioned amping up disinfection between sites would be a good biosecurity response. So
Yeah, that's great. Yeah, very important.
and I think they'll have a paper coming out within the wildlife professionals sometime this winter, kind of about that topic as well. And we have a question from Jonathan Colby. What would be the best way to disinfect hundreds of Ranavirus contaminated cardboard boxes at a time? Bleach, incineration, drying, thinking about bullfrog trade and biosecurity.
That's a question I've never had before, but I would guess that incineration would be the easiest way to go, but I'm not sure.
Dee Dee mentioned it is the December 2023 issue of the Wildlife Professional that will have that article. So keep an eye out. If you get it, if you don't, consider subscribing.
I think it's important to mention that, you know, I think there's no evidence that the DNA, the viral DNA is infectious. Yeah, it has to be in a capsid. So as long as the, you know, the capsid has been damaged by bleach or something like that, or we have here, we use another disinfectant, I forgot the name, but, you know, one of those chaotropic agents, that should be plenty enough, I think.
Yeah, I think if you can soak the cardboard in like bleach or something, like that would work. It just maybe incineration is easier.
Yeah, if it's a big place, yeah.
All right. Well, I don't see any more questions in the chat, and we are at 12:35, so we're just a couple minutes over. So I want to thank all of our speakers again for being here today and giving us your time to present on all of this. We really, really appreciate it. And thank you to the Global Ranavirus Consortium for continuing this partnership and helping to have this webinar series together. So this is being recorded, and we will be putting it up on the PARC YouTube channel once we have the closed captions finished with all of that. And so those of you who registered,
I can just add, remind again people, so the book, the revised book, Ranavirus book will be soon, you know, we are close to make the things finish. And again, when published, it will be available for everyone. And I think that's a good update for people interested. And then don't forget that we have then the scientific session coming later on at 2 p.m. Yeah.
Yep.
Maybe
and I will, is it okay if I drop those links in the chat?
Yeah, exactly. That's why, I was thinking maybe because maybe we didn't get everybody.
Yeah, if you didn't get them, I just dropped those links in the chat for this afternoon session. So, so yeah, you have got some great other talks scheduled for Ranavirus, including one that we had a shout out for in the chat. So.
Yeah, and feel free to share those links with people you think would be interested. It's not a closed session. It's just we shared the link with people who registered, but happy to spread that to more people.
Right.
Okay, thank you so much.
Thank you very much.
Bye, everyone.
We'll see you all